aliphatic and aromatic carbon rings effortlessly.
So just finding organic carbon on Mars or in the plumes of Enceladus is fully expected. It's not a biosignature at all.
Not even close.
So if a rover drills into a dried-up lake bed like Jezero crater on Mars and its mass spectrometer detects organic compounds, how does the science team determine if it's just the chaotic remnant of a meteorite impact or if it's actually the degraded remains of an ancient cell wall?
Well, the distinction really comes down to intent, like organizational intent.
Okay, explain that. How does a molecule have intent?
Abiotic chemistry is chaotic. It's thermodynamically random. If you rely on deep space radiation to synthesize molecules, you get this sprawling, disorganized mess of different shapes, sizes, and branching structures.
A random assortment.
Exactly. But biology is a relentless optimizer. It is highly, highly selective.
It acts like an engineer. It doesn't use every material available. It finds a blueprint that works efficiently and just mass produces it.
Exactly. And the most pristine example of this biological selectivity is something called chirality, or molecular handedness.
Handedness. Okay, I love this concept. Walk us through it.
So many complex organic molecules, including amino acids, are chiral. That means they can form in two distinct structures that are non-superimposable mirror images of each other.
Much like a left and right hand.
Precisely. They have the same atoms, but mirrored. Now, if I synthesize a batch of amino acids in a sterile laboratory without using any biological catalysts, what do you think the distribution of left-handed and right-handed molecules will be?
Well, based on the laws of probability, I'd assume it's a 50-50 split.
Yes, a racemic mixture. The chemical pathways have absolutely no preference for one orientation over the other, so the universe basically just flips a coin every time a molecule forms.
But biology doesn't flip coins.
No, biology rigs the game entirely. Every single living organism on Earth, from an archaea at a deep-sea hydrothermal vent to a blue whale, utilizes exclusively left-handed or levodopa amino acids to construct its proteins.
Every single one. That's insane.
Every single one. And conversely, biology exclusively uses right-handed sugars for its nucleic acid backbones.
The implication of that is just staggering.
It really is.
So if our rover on Mars pulls a sample of regolith, isolates the amino acids, and measures the chirality, finding a 50-50 split means we're just looking at cosmic dust.
Right. Abiotic chemistry.
But if we find an enantiomeric excess, say a sample where 90% of the molecules are left-handed,
Then you have found the fingerprint of an architect. The thermodynamic laws of the universe do not spontaneously sort molecules by their handedness.
they just don't do it.
Never. An enantiomeric excess of that magnitude requires a catalytic system that actively selects and processes specific chiral structures while discarding the rest.
It's like finding a molecular brick wall laid with absolute precision instead of just a random pile of bricks.
Exactly. It completely defies the entropic chaos of non-living chemistry.
I love that brick wall analogy, but we aren't just looking for microscopic molecular brick walls, are we?
No, we're not.
If we zoom out from the mass spectrometers and just use the rover's optical cameras, are there physical macroscopic structures we can actually see that prove life existed there?
Yes, and this is the pursuit of structural biosignatures. But we have to heavily temper expectations here.
Right, like we said earlier, we aren't traversing the Martian landscape looking for dinosaur bones.
Exactly. The most ancient, durable records of life on our own planet weren't made by macrofauna. They were constructed by microscopic organisms long, long before multicellular life even evolved.
We are talking about microbial communities altering the physical geological record.
Yes, and the source text focuses heavily on something called stromatolites.
Stromatolites, which are essentially the fossilized behavioral patterns of bacteria, right?
That's a great description. Stromatolites are laminated, accretionary structures formed by biofilms of microorganisms, primarily cyanobacteria.
Okay, so how do they actually form? Because it's a really fascinating fight for survival, isn't it?
It is a relentless, repetitive battle against burial. So picture an ancient shallow water environment. These bacteria form dense, sticky mats on the sediment surface.
To maximize their exposure to sunlight, right?
Yep.
Photosynthesis.
Exactly. But then the tides wash in, and they carry suspended sediment, silica, and calcium carbonate. That dirt settles directly over the bacterial mat, effectively burying them.
And if they remain buried, they're cut off from the sun and they die.
Right. So they exhibit a phototactic response. The entire microbial community literally grows upward, migrating through that newly deposited dirt layer to reestablish a new mat on the surface.
So they essentially bind the sediment, cement it in place, and climb over it.
Over and over again. Over millennia, this cyclical process of burial, binding, and upward migration produces distinct, finely layered macroscopic rock mounds.
Wow.
The biological tissue eventually decays, of course, but the mineralogical structure, the architectural footprint of their struggle, lithifies into stone.
Which explains exactly why NASA landed the Perseverance rover in Jezero Crater on Mars.
Yes, Jezero Crater is the site of an ancient Martian lake. It has a highly preserved river delta.
So if shallow water microbes existed on Mars billions of years ago, the margins of that crater are the absolute prime real estate to find stromatolite-like structures.
They are. But the challenge here, similar to the atmospheric false positives we talked about, is morphological mimicry.
Mimicry, so abiotic rocks pretending to be biological.
Basically, yes, abiotic chemical processes can create layered rocks that look deceivingly like stromatolites. The Apex Chert deposit in Western Australia is a classic example of this controversy.
Oh, I've heard of that. People argued over it for years.
For decades, researchers argued endlessly over whether the microscopic filamentous structures found in those rocks were the oldest fossilized bacteria on Earth, or if they were simply complex chemical gardens formed by hydrothermal fluids precipitating silica.
So if Earth geologists couldn't even agree with the rock sitting right in front of them, how does a rover on Mars prove that a layered rock isn't just an abiotic chemical garden? It can't just take a picture and call it a day.
Definitely not. It requires multimodal analysis. You don't just look at the morphology, the shape. You have to integrate the chemistry.
Okay, how does the rover do that?
The Perseverance rover is actually equipped with an instrument suite called Sherlock.
Ah, yes. Scanning habitable environments with Raman and luminescence for organics and chemicals.
Very good. They really, really tortured the acronym for that one.
They absolutely did, but the technology is revolutionary, isn't it?
It is incredible. Sherlock uses a deep ultraviolet laser spectrometer to map the spatial distribution of organic molecules on the surface of the rock.
So it can see exactly where the organics are located.
Yes. So if we find a layered structure that looks visually like a stromatolite, and then Sherlock confirms that complex aromatic organics are tightly clustered strictly along the specific layers of the rock, correlating perfectly with the visual laminae, the argument for biology becomes incredibly robust.
We are cross-referencing the structural morphology with the chemical architecture, which, if we connect this to the bigger picture of the scientific method, just shows the sheer rigor required to officially declare a biosignature discovery. It's immense.
It has to be immense. The astrobiology community is, well, they're haunted, in a productive way, by the Viking lander missions of 1976.
Oh man, the Viking missions, that was a roller coaster.
It was. The Viking probes conducted metabolic experiments on the Martian soil, and one of the experiments, the labeled release test, actually yielded highly positive results.
It looked remarkably like biological respiration, right? Like something was eating the nutrients and breathing out gas.
It looked exactly like that. But the scientific consensus today is that those results were entirely abiotic.
Why? What fooled them?
It turned out the Martian soil contained highly reactive chemicals called perchlorates. And these perchlorates mimicked the metabolic release of carbon when they were exposed to the nutrient broth the lander used.
And the Viking payload just wasn't equipped to identify the perchlorates.
Exactly. Yeah. Which led to decades of debate and confusion. We learned the hard way that ambiguous results are the default when exploring alien geochemistry. The burden of proof for the next discovery is unprecedented.
We really have to be sure. Let's shift our gaze back to the cosmos for a minute, because our modern toolkit for atmospheric detection has also received a massive upgrade recently.
Oh, a huge upgrade.
We discussed transit spectroscopy earlier, and right now, the James Webb Space Telescope is currently executing this exact science on the TRAPPIST-1 system.
Yes, TRAPPIST-1 is a cornerstone target for astrobiology.
Remind us what TRAPPIST-1 is.
It's an ultra-cool red dwarf star located roughly 40 light-years away from us. And it hosts a very compact system of 7 terrestrial rocky planets.
And several of these planets orbit right within the empirical habitable zone, right?
Exactly, right where insulation levels theoretically allow for liquid surface water to exist.
But observing an M-dwarf system like TRAPPIST-1 introduces a whole host of violent variables that we just don't experience with our own sun.
We don't. Red dwarfs are notoriously active, especially in their youth. They unleash massive stellar flares and coronal mass ejections constantly.
And because the habitable zone around a dim star is so physically close to the star, those planets are just getting hammered.
They're subjected to intense, extreme ultraviolet and X-ray radiation.
Which raises a really critical question about atmospheric retention. Like, Could those flares just blast the atmosphere away entirely, leaving a totally sidereal rock?
That is exactly what JWST is trying to determine right now. Before we can even look for biosignatures, we have to prove these planets haven't been completely stripped bare.
Have they found anything yet?
Well, early thermal emission phase curve data from TRAPPIST-1b, which is the innermost planet, suggests it lacks a significant atmosphere.
So it probably got blasted away.
Likely, yes.
Mm.
But if we look at the planets further out in the habitable zone, like TRAPPIST-1m or F, and JWST detects a robust secondary atmosphere there.
Then the hunt for disequilibrium begins.
Exactly. We stare at those transit spectra, gathering precious photons for hundreds of hours, looking for that impossible pairing of oxygen and methane.
While simultaneously trying to model every conceivable abiotic photochemical pathway that the red dwarf's radiation could trigger to fool us.
You've got it. It's a daunting task.
The level of caution is necessary, obviously, but we also have to recognize the stakes here. So what does this all mean for us? You and me, the listener, humanity.
Okay.
We are investing billions in orbital observatories. We are sending nuclear-powered rovers to vaporize rocks on other planets.
It's a massive endeavor.
The technical execution is fascinating, but the philosophical payload of this deep dive is what truly alters our worldview, I think.
I agree completely, because currently our sample size for biology in the universe is exactly 1.
Just us.
Just us. The genesis of life on Earth could be the result of a highly improbable, statistically miraculous sequence of events that has never been replicated anywhere else in the galaxy.
Right, the rare Earth hypothesis. The idea that we are just a bizarre, localized fluke in an otherwise cold, sterile cosmos.
But the moment we detect a definitive biosignature, whether it is a chiral excess in a Martian dirt sample or an inexplicable oxygen-methane standoff in the atmosphere of an exoplanet 40 light-years away,
that entire paradigm just collapses.
it shatters.
It transitions life from a statistical anomaly to a thermodynamic imperative.
Exactly. It proves that biology is a fundamental emergent property of the universe. When you combine liquid water, organic carbon, and a stable energy gradient, the chemistry doesn't just stagnate, it inevitably reorganizes itself into complex, self-replicating systems.
It implies that the universe is aggressively wired for life.
It does.
Building on that, I want to leave you with a final, slightly provocative thought to chew on today. If the laws of physics and chemistry dictate that matter inevitably awakens into biology given the right conditions, does that imply something deeper about the architecture of the cosmos?
Oh, that's a profound question.
Because if consciousness is the eventual evolutionary endpoint of that biology, then we are quite literally the universe developing a nervous system to perceive itself.
Wow.
If life is a built-in feature rather than a random bug, perhaps the night sky isn't a silent void waiting to be explored. Perhaps it is a vast, interconnected chemical network that is slowly, over billions of years, waking up across billions of worlds.
That is a staggering realization to carry with you the next time you look at the stars.
It really is. So the next time you look up, don't just see cold space. Look at those twinkling dots and recognize that the light hitting your eye is carrying encrypted secrets about the thermodynamic state of distant worlds. The cosmic blueprints are out there whispering across the vacuum. And for the first time in human history, we finally have the tools, the physics, and the scientific rigor to read them.
We really do.
Thank you so much for joining us for this deep dive at Stellar Lab. As always, we appreciate your curiosity. Keep looking up, keep asking questions, and we'll see you next time.